Hi everyone, here we have Mihail Todorov from my lab, a PhD student.
Hello.
Who has been operating the LaVision microscopes since last a couple of years and also has a good technical knowledge on the microscope. So I would like to ask him to demo you in particular how to place samples onto different sample holders. In my lab, now I guess more and more labs are interested in imaging larger samples. However, there is not much option as a sample holder how to place the large sample to tissue. And Mike has some solutions who wants to share with you. So Mike, can you show us, for example, what are the options to image larger samples, large part of the mouse or whole mouse, how one could image using the system?
Sure, thank you, Ali. So here I would like to present you some of the sample holders that we have in place. We are using actually every day. Just a brief mentioning that depending on the sample size, you can see from really small samples that can go via 3D printed sample holders.
So those are used for slices?
Brain slices, usually, yeah. We can have various different sizes depending on the tissue, of course. Up to large scale holders, also here you see a sample actually mounted, up to really big that we use for also custom made for scanning the whole mouse intact without sectioning. Now I would like to show you how the sample has to be placed into the microscope so that we can start the acquisition.
So after clearing the tissues, of course, then the sample is mounted, and usually that may require fixing with some wires or gluing, and we usually now use mostly gluing, I think.
Yes.
And then here we have about half body part of the adult mouse. Mike will show you how to put into the microscope and generate some images.
Okay, so now basically when we have the sample straight and fixed to the holder, we can submerge it into the liquid. The very interesting part is that if you leave the laser on while you are doing the dipping, you can actually see and make sure that all your clearing and labeling has worked, because even with the naked eye you can see all the Z slices being optically made by the laser, by the very thin light. Whole point of the acquisition is actually that after calibrating the system, which is very important because now we are really talking about micrometer thin light sheets scanning through the sample. Of course, the right and the left one has to be really well aligned. For this, of course, the company provides very good tutorials that you can check out in combination with the special alignment tool that they have. So after putting the objective in the light path through the revolver, we can start actually dipping the whole system into the imaging solution, which will give us unrestricted access to all the microanatomy that we have. So first we position the sample in a way that we see the very top of the sample, that we can be sure that we will not hit it with the objective while descending into the liquid. And we slowly have to see with the eyes when we are disturbing the surface of the liquid, then we know we are in, as now. And basically, then we need to find the sample using the correct focusing, as seen on the screen. And because we are talking about micrometer precision or submicron precision, actually we need, from this point, we don't need to touch the setup. We are just using the keyboard and the controls, the motorized controls of the microscope. So using a background channel in whatever spectra is available in the fluorescent channels, we can easily now slice through the sample.
Okay, so now Mike placed the sample into chamber and operating the microscope to show us how one can use this software to image the disco cleared samples. And here again we have an upper part of a whole mouse that is cleared with disco technology, and we have labeling of vessels, and we have labeling of cell bodies. We have two different channels. Now we are looking into the mouse set, I guess. So, Mike, can you please tell us more about the sample?
Yes, so this particular sample has been stained with dye to mark really all the blood vessels, as you can see through the complete intact tissue up to the capillary level. I will just show you in a minute. And also we have a counter staining based on propidium iodine, which stains really all cell bodies in the intact tissue. Let me just zoom in quickly so that we can see the sheer quality actually. For example, let me see. It's a bit of zooming in game. But here each and every dot represents one single cell. But the good thing is we are considering here the skull, intact skull structures, but as you can see also in the brain tissue, we really have truly micrometer scale resolution.
These are cell bodies on in the skull part.
Cell
as well
nuclei.
as in the brain.
Yes.
Nuclear labeling.
Yes.
Propium iodine. And how about the vessels?
In the vascular case, we also have very convenient staining. So
Using lectin dye.
yes, lectin is the golden standard actually to really be able to acquire all the continuity of the vasculature.
Mm-hmm.
Let me maybe even show more of the data, not too much. Yeah, but frankly with the very advanced camera, we can really, as you see, capture the golden standard 16-bit data, which is then very useful for machine learning based segmentation and to get any quantitative data from it.
Cool. And maybe another one of the last things, how easy to operate the software? So can you show the main functions, what has to be clicked and so on?
So the very important point of the software is actually where you start and stop the acquisition. Here you can also set up the different focusing strategies. Convenient part of the software is that you can set the laser power or the emission light intensity without actually touching the microscope, just by digitally adjusting the different settings here. We have different filters as in the traditional fluorescent microscopy, and we can adjust the direction of the light or either use both light sheets at the same time that we get a really even illumination. This we usually do for the acquisition. And then we have like traditional Z-stack ranges that we can set, and also the Z step size as well if we like to have multi-volume acquisition to later reconstruct the whole animal. Then we can set up a tiling scan that can be then later reconstructed using computer science tools. After we start the acquisition, first we make sure that our tissue is well stained homogeneously. In this case, we do an overview scan, which takes about half an hour maximum. And then we start the real, very precise measurement, which makes sure that we capture the whole continuity of vessels and each and every single cell.
Mm-hmm. And that would take maybe some hours, a day, or two, depending on the sample.
Yes, of course, depending on the sample size.
Cool. Good. So how about looking at the sample, looking into really where is the sample and how light passed through?
Yes, this is very convenient as well. So even at the first time that you finish clearing and labeling your sample, you can be sure by switching on the laser and putting your sample inside the microscope, you can actually see how it's been on the fly, real-time sliced through the light.
Mm-hmm.
So if you move it here like this, you can actually observe each and every Z-slice that you're going later to acquire. An important step using this immersion liquid for imaging, we really need to make sure that there are no air bubbles or other types of gas bubbles building up, because they prevent the legit acquisition of the signal.
Okay, how do you get rid of the air bubbles?
To get the air bubbles, usually it's
So we are
actually
talking about now organic solvents, which is potentially having the list of problems, because I know other clearing solution using sugar-based immersion media is much more bubbles. Okay, in organic solvents, if there's any, so
it's
what do you
very
do?
rare. I would say that it's not really often, but sometimes in critical samples you need really to play a bit with the sample. Usually just really moving it a bit in the liquid does get rid of the sample.
Okay, so you're talking about air bubble in the in the sample.
Trapped,
And then you
yes.
try to
You can
fill
also
it with
dip
the
it a
solution.
bit or like
Okay,
tilt
so
it.
I got it. Cool.
And this makes sure that really you have a 3D correct model of your data.
Mhm.